Okay, here we are with Chapter 18, or the video lecture for Chapter 18. I may get this one all done in one video lecture. Not sure. We'll see how it goes. It may be two, but two would be the most. All right, and again, this is going to be the last chapter for the semester, okay? So here in this chapter, we're going to talk about speciation. Speciation is defined as the evolution of reproductive isolation within an ancestral species that then results in two or more descendant species, okay? So the key point here is reproductive isolation. And a critical question with regard to speciation then, or at least in terms of understanding how speciation occurs, is to understand how genetically based reproductive barriers to gene flow come about. What kinds of processes can cause genetically based reproductive barriers to come into existence or to kick in. So to understand the problem, let's consider two sort of hypothetical populations in which different alleles have become fixed. So let's say in population 1, we have allele A1 fixed, so that all individuals therefore are A1 homozygotes. They all have that genotype. In population 2, let's assume that A2, or allele A2 rather, is fixed, so that all individuals there are A2 homozygotes. They all have that same genotype. The question is, how could these two populations have diverged so that different alleles became fixed in each? So think about it. If allele A2 arose initially in population 1 as a new mutation, where in population 1, A1 was close to fixation or was fixed, allele A2 would have had to be present in a heterozygote. If it was just one copy of allele A2 in a population where all the other alleles were A1, then the individual who had A2 as a new mutation must have been a heterozygote, an A1-A2 heterozygote. Now, if that heterozygote, as is so often the case, had low fitness, there is theoretically no way that A2 could have increased in frequency, forming an A2-A2 population that is then reproductively incompatible with the A1-A1 population. How could A2 increase if it's initially present in just one copy in a heterozygote, and heterozygotes are being selected against? It doesn't seem to make sense. The other problem of explaining speciation is that let's assume heterozygotes or intermediate phenotypes are actually fit. If they are fit, they would then constitute a bridge for gene flow between two populations, and this would then prevent those populations from diverging genetically. They would have gene flow from one gene pool into the next, and eventually they would just homogenize. So these are two problems that we have to be able to work around or find solutions to in order to understand how genetically based reproductive barriers can come into existence. So these two issues then can be summarized in the following question. How can two genetically distinct and reproductively isolated populations form without intermediates or without heterozygotes? Well, the paradox then is that fit intermediates are necessary for speciation, i.e. to fix a second allele in another population, you have to have fit heterozygotes, but their very existence would seem to prevent the two populations from diverging in the first place. That's an apparent paradox. Okay, so let's talk about modes of speciation. Then as we go throughout this discussion, we'll see some possible answers to or solutions to that paradox. And the solution ultimately lies in the various ways that speciation can occur. Okay, so these are the modes of speciation we're going to discuss. All right, the first mode of speciation that we're going to discuss is called allopatric speciation. All right, allopatric. And I think I mentioned allopatric in the previous chapter. Allopatric means other country. So in allopatric speciation, you have a geographic barrier that prevents gene flow, resulting in reproductive barriers as gene pools follow their independent evolutionary trajectories, either through selection or drift or some combination of the two. So here, gene flow is reduced initially by a physical barrier, such as like a desert, a river, a mountain range, etc. Okay. Second type, and we'll see more detail on that in a little bit. The second mode of speciation is referred to as parapatric speciation. Okay. Here, we have populations that are geographically adjacent to one another that do not interbreed at the zone of contact and thus evolve into different species. We mentioned that a little bit in the last chapter with hybrid zones. All right, third, we have what's called sympatric speciation. So sympatric means same country. Here we have reproductive barriers that evolve within a single, initially panmictic population. Panmictic means randomly mating, okay? So you have a population that's spread over a geographic range. They can all encounter each other. They can all freely mate with each other, but you still have reproductive barriers that evolve that eventually split at least one part of that population into a separate species, even though they would appear to be able to mix their genes very well. All right. In this case, in the case of sympatric, initial isolation occurs through the evolution of some biological feature of the organisms themselves. It could be female mate choice, as one example. But because this one's less cut and dry than either allopatric or parapatric, sympatric is actually quite controversial. There are some evolutionary biologists who don't view it as even being a real phenomenon. Okay, let's look at some examples of these. So this figure is from the textbook. So on the left here in part A, we have allopatric speciation, and this says by vicariance. Vicariance refers to some physical barrier that literally splits the population into two separate populations. They physically can't get past the barrier, like a river or a mountain chain or a desert or something like that. So let's start with an initially continuous population. A barrier occurs, maybe a river changes course or cuts down below a certain level in the rock or whatever. But anyway, the population is ultimately separated, and this is a barrier to gene flow. And as long as they're separated long enough, they'll begin to evolve in separate directions because drift is doing different things in those populations. Selection might be operating differently. They will start to diverge genetically. The gene pools are becoming different. And if enough time has gone by, they can eventually become reproductively isolated so that even if they do happen to come together at some point again in the future, the barrier goes away. Now there are genetic barriers to gene flow, pre-zygotic, post-zygotic barriers like we talked about in the last chapter for example, and that would keep those two populations separate genetically. And if they're different gene pools which don't intermingle, then we know according to the biological species concept, they're defined as different species. Allopatric is the most cut and dry, the easiest to understand in terms of how this might happen. A great example from my own research that extended from my doctoral dissertation. I was working with cavefish that were found in caves throughout southern Indiana and across the Ohio River into northern and north central Kentucky. And initially they were thought to be one species. Well, the Ohio River, which is a major river, runs right through the middle of their distribution. So I had hypothesized that if we looked at genetic data, we might see that there are actually two different species. Now, cavefish populations have been there, we estimate, probably for several million years in those caves. The caves aren't much older than that, we know. The Ohio River, as it currently exists, is only about 800,000 years old. That river formed about 1 million years ago or so, 800,000 years ago, as a result of some changes that occurred due to the last glaciation up in New England area, in Pennsylvania in particular. And the river became much bigger than it was in the past. It used to just be a small stream. Well, once that river became much larger, it began to cut much more deeply into the bedrock and actually cut down through the level of the cave-bearing strata in southern Indiana and northern Kentucky, and eventually separated those populations into two different groups. And cavefish cannot get across the Ohio River. They're just not adapted for it. They're easy pickings for a lot of other predatory species. So my hypothesis was that when the Ohio River became much larger, about 800,000 years ago, cut down through the bedrock, it allopatrically separated the northern and southern populations, and they have since been evolving into separate species. Well, we took mitochondrial DNA samples and basically confirmed that very thing in a paper that was published, about 8 years ago now, I guess, in the journal Evolution. So that was a great example from my own research of allopatric speciation. We know it occurs. All right. We're going to come back to B here in a minute. Let's go on to C. C is parapatric speciation. So here we have a continuous population. Maybe there's an environmental gradient like temperature, altitude, whatever, right here in the middle, and that results in a cline. Remember, we talked about clines, right? You can have an allele that changes frequency rapidly at that environmental boundary, and that's a cline. And if that persists long enough, you can have populations on either side that essentially, or eventually rather, become genetically isolated from each other, as we've seen in the last chapter. And again, the longer it goes on, the more isolated or genetically distinct they become, and eventually you might get a hybrid zone forming between them, but that can be a tension zone like we talked about in the previous chapter. Still, you can have separate species evolving on either side of it. So what you get then is two populations whose distributions, or two species, I should say, whose distributions do actually come into contact, and you may or may not have a hybrid zone there, but you have two different species whose boundaries or borders of the geographic range do sort of butt up against each other like that. But they evolve from the same ancestral species. Here we have sympatric speciation. So we have a population that's panmictic. All these individuals of the same species can intermingle and intermate and mate with each other. But let's say that for some reason a mutation in the DNA causes a new male to form, one with a different color pattern, let's say. And let's say there happens to be a female somewhere in that population with a genetic constitution that makes her very attracted to that new type of male. They might start to mate preferentially with each other. They produce offspring, who also then would sort of cause that new population of individuals who look different as males and have different preferences as females to sort of isolate, self-isolate. That's an apropos term, isn't it? Here within the same population. And eventually you get two new species, or two different species. One new species evolving from the ancestral species. There's no physical barriers whatsoever. It's not allopatric, it's not parapatric. You have a new species evolving from an ancestral species right in the midst of the range of the ancestral species. Again, that one is somewhat controversial. If it does happen, it's thought to be more prevalent in plants because of some of the ways that they can reproduce. For example, self-fertilization, which animals typically can't. Now let's go back to B. B is called peripatric. Note that's different, right? We have para here and peri there. Parapatric speciation is also called founder effect speciation. So the idea here is we have a continuous population, a small group breaks away as a founder group for a new population. And again, because they're a very small population, most likely, their genetic constitution, their allele frequencies and genotype frequencies are probably going to be different because of drift from the parent population they came from. They start off on a new genetic footing. Maybe they're in a different area now and they're adapting to different conditions. They pretty rapidly evolve into a new species. They could come back together or could sort of grow, the population grow and come back into contact with the ancestral population. But now they're different enough where there's no intermating between them, and so you have two distinct species. So the range expansion there could reestablish sympatry, an area of overlap between them, but now there'll be two separate species. Okay. So these are the various mechanisms through which we think evolution, or speciation rather, occurs. All right. So again, allopatric speciation, which is probably one of the more common types and the most straightforward in terms of understanding the dynamics and how it might work, is going to require some sort of physical barrier to separate a contiguous population into two separate subpopulations, at least two separate subpopulations. Then you get, as a result of that barrier, like the Ohio River, for example, a restricted or non-existent gene flow between the two separated populations that allows for each of them to develop genetic differences so that if the populations ever do come into contact again, they might now be genetically different enough that they're unable to exchange genes. Okay, so what kind of evidence do we have for that, for this idea that geographic separation causes populations to diverge genetically? Well, there's lots of evidence of it. I mean, I talked about evidence that I found with my cavefish species we were looking at. By the way, those have now been classified as two separate species as a result of that work. Here you see an example with dusky salamanders. I believe this data was taken from Western North Carolina. The Smoky Mountains National Park area. That's actually a hotspot for salamander diversity. It's one of the highest, the areas of highest salamander diversity on the planet is here in western North Carolina. Anyway, what you're seeing here are different populations. These different points represent different populations of the salamander. Here you have geographic distance in kilometers, and here you have sexual isolation index, which is a measure of how likely or how successfully, I guess is a better word, individuals from one population are to mate with individuals of another. So what happens is that as geographic distance between populations increases, the sexual isolation index also goes up. That means that populations that are, 250 kilometers apart from each other are going to have a fairly high isolation index. Individuals from those populations, when they're put together, just don't mate, or if they do mate, they don't reproduce very successfully. That's suggesting that geographic isolation between them is causing or allowing genetic differences to evolve that then prevents successful reproduction. That's the basis of what allopatric speciation is and what's occurring in allopatric speciation. Here we see a measure of genetic distance right here. So very similar to very different. What we see is that as genetic distance goes up, as we see differences evolving between, or genetic differences evolving between populations, their sexual isolation goes up. Okay, so that's saying the same thing. It's saying that the more geographically separated populations are, the more genetic distance there is between them, the more sexually isolated from one another they are. And that's all support for the allopatric mode of speciation. All right. Other evidence. Historical barriers can actually be inferred from spatial patterns of DNA sequence differences. So, barriers that were present in the past that are no longer present could leave a mark in terms of their existence in modern species. And here we see evidence of that. So here we have 6 different species of, or southeastern species of freshwater fish. So spotted sunfish, warmouth, bluegill sunfish, red ear, bowfin, and mosquito fish. And you see these are all southeastern species, southeastern coastal plain species. And what you see in every single one of these 6 species is 2 separate clades. A western clade, shown by the orange populations, and an eastern clade shown by the black populations here. Okay. In most cases, western Florida seems to be the divide between them, or at least a line, a longitude along that point. Turns out genetic analysis has indicated that whatever it was that caused these populations to diverge happened about 3 or 4 million years ago. And it turns out about 4 million years ago, there was a shallow inland sea that covered, much of Georgia, Alabama, Mississippi, right in here. And so these rivers didn't exist. So you had these western clades, western groups over here, sort of towards Louisiana, Arkansas, Texas area, that were separated from the eastern groups here by that inland sea. When it went away, they then sort of came back together, and now we see evidence that they have evolved separately while that inland sea was present. That's why we have 2 separate clades today. That's historical evidence for allopatric speciation. Other evidence for allopatric speciation comes from small islands which lack any obvious geographic barriers, right? Very often small islands don't have barriers. They're just too small. Because of that, islands typically do not harbor closely related species, what we call sister species, of birds, for example, because they're not large enough to present reproductive barriers. So when 2 or more closely related species do actually co-occur on an island, in other words, are sympatric on an island, evidence suggests that one species probably migrated from another nearby island where it evolved in isolation. Obvious geographic barriers would include oceans, rivers, canyons, deserts, those sorts of things. But what about the many examples of species diversity within continental interiors where isolating geographic barriers like that are often absent? There, because you have an absence of physical barriers, it's thought that climate and maybe suitable habitat although not obvious barriers, could be just as effective at isolating a population as a river, a canyon, or an ocean might be. This brings us to the concept of what is called climatic niche space. Okay, climatic niche space. This is a geographic region that's delineated by specific climatic and habitat requirements of a given species, outside of which survival declines. That leads to what's called niche conservatism. Niche conservatism. In other words, being dependent on specific climatic or habitat conditions. And this is hypothesized to actually promote allopatric speciation. There's some evidence for this, especially from lungless salamanders, the plethodonid salamanders. So allopatric sister species of lungless salamanders occupy regions with very similar climatic conditions and are often absent from areas with unsuitable climates. Okay, that's the climatic niche space idea. Sister species are also absent from much of each other's range, even though the climate is suitable for both species. And this pattern suggests that they cannot get past some sort of geographic barrier between them. There is evidence of this, again, from western North Carolina, again, where we have this hotspot of salamander diversity. And so what you're seeing here is the geographic range of many of these, or some of these plethodonid salamanders. We have Plethodon cylindricus, Plethodon teahali, and then we have an area shown in orange where the climate is actually suitable for both. So the yellow area here represents the climatic niche space of Cylindricaeus. This is where it can actually persist. The purple area here is climate or areas suitable for Plethodon teahali. That area in orange here and here is an area, or areas rather, where the climate is suitable for both species. But if you look, notice that the orange dots are teahali and the black are Cylindricus. You find mostly the orange teahali species here. On the other area where both species can persist, you find mostly Cylindricus, all right? That suggests there's some sort of barrier in between them there that is separating them, okay? But the thing is that we don't actually see that today. So if we look down here, for example, this is sort of showing you in part B of this figure what may have happened. It could have been that the ancestral species with a specific cold tolerance was found at lower elevations. That's the white band there, right? And it straddles or actually encompasses this valley between the two peaks here. Climatic warming over time may have caused the distributions of these species to actually rise up the mountains, because as the environment gets warmer, their cold tolerance range shifts up, okay? They can't survive down here anymore because it's simply too warm, and so they have to actually move up the mountain as the climate warms. Once that happens, eventually they move high enough where the base of that valley is no longer in their range of cold tolerance and they can't get across it anymore. So effectively they've been separated into two allopatric populations by its shift in the climate. And so it's thought that that may have been actually what happened. And so today we have two different species that both occupy areas that both species can actually exist in terms of climate, but they're separated by this valley here, which now represents an area where their tolerances are simply not, or at least the tolerance of one is not geared toward it. They can't survive there. So they've been separated, and who knows how long this has taken, but they've been separated by probably thousands of years of time by a past climatic change event that forced their distributions higher up to those towards the peaks of those mountains and past that valley, which would have represented a pathway for gene flow. And now that's been cut off to them because of climate change. All right. Now, vicariance, again, as I mentioned before, is separation of populations as a result of geographic barriers. And most evolutionary biologists view allopatric speciation through vicariance to be a consequence of natural selection within the isolated populations, causing prezygotic and or possibly postzygotic reproductive barriers to evolve. So these populations are separated. There's no longer gene flow between them. Selection and drift are operating on them independently, making them become or diverge genetically. And so you get prezygotic and or postzygotic reproductive barriers, so that if they do ever meet again, if the barrier goes away, now their reproductive modes are simply no longer compatible. All right, so they had reproductive barriers that evolved while they were allopatrically separated. Okay, let's talk about peripatric speciation now. So peripatric speciation, again, P-E-R-I, Patrick, let's clarify that. This is again called founder effect speciation. And so this concept of peripatric speciation was introduced by actually a famous evolutionary biologist named Ernst Mayr. Ernst Mayr suggested that a small group that was cut off from gene flow with the main population of the species could undergo rapid genetic changes due to genetic drift. Remember, we said the effect of drift is higher the smaller the sample is. So the smaller the founder population, or the population that migrates away from the parent population, is, the more pronounced the effect of drift would be, the more different they would be in terms of genotype and allele frequencies from the population they came from. And so Mayr proposed that due to such sort of accidents of sampling, in other words, genetic drift, allele frequencies would differ from those in the parent population, and that would provide a different genetic starting point for the population to evolve from. What kind of evidence do we have for this? Well, small islands. So this is the island of New Guinea in Southeast Asia, okay, or actually just north of Australia. And what you see is that we have various species of kingfishers. So here you see T. galatea, T. carolinae, and T. riadelii. Now, some of these have fairly large distributions, as you can see, but some of them are also found on these very, very small islands. And it's thought that initially we probably had maybe one population or one species of kingfisher maybe on the large main island of New Guinea, and then as some of them began to migrate out to some of these very small islands, which in some cases are pretty far separated from the mainland, that was an example of founder effect speciation, or peripatric speciation. So these populations evolved into new species possibly fairly rapidly as a result of that founder effect population being very small, drift causing its allele frequencies to be quite different initially, so it's sort of immediately on a different genetic starting point or different genetic footing. And then it moves to a new island where climatic conditions might be different, there are different food types, different plant communities, for example, and selection takes over and just takes that population in a new direction very rapidly. So we do see some good examples of evidence that suggests that this may actually be a real thing. All right. So the initial alteration of allele frequencies due to drift is what Mayr, again, called the founder effect. And the principal idea here is that genes often work together epistatically. One gene would affect the expression of another to affect fitness. So shifts in allele frequencies at some loci may affect fitness of certain genotypes at other epistatic loci, right? Epistatic means different genes work together. So changes at these loci may then cause epistatic changes at yet other loci. So you'd have this sort of snowball effect that could result in fairly rapid reproductive isolation. Now, Mayr argued that such evolutionary changes may take place so rapidly that they might not be even recorded in the fossil record. They happen so quickly that the fossil record doesn't have that kind of resolution to show that sort of thing. And this would help to explain why transitional forms between species are often rare in the fossil record. And if you remember, we talked about the concept or the theory of punctuated equilibrium that Eldridge and Gould put forth in the 1970s. Meyer's concept of founder effect speciation and its implications for the fossil record are actually embodied in that model of punctuated equilibrium. Okay, so I think that's actually a good place to stop. The next thing we're going to get into, I guess, which is going to be the second part of this video lecture, is back to the idea of adaptive landscapes. So we'll get to that in the second part here.